Hello, Chile from China. I have 15 minutes to tell you a story. What is precision medicine? What can we do about it? And what's the challenge? Oh, too fast. Why do we need precision medicine? That is because we are not precise when we take medicine right now. Secondly, I will tell you why we are not precise in getting medicine. That's because all the research work we have been doing over the past decade have not been reproducible. We use all the omic technology. I will talk about what is omic technology later. And thirdly, I will share with you what we are doing in terms of quality control, standardization to make precision medicine not only a dream, but a reality. You can read this figure. Most people taking the drug, I think they believe they will get cured by the drug. But unfortunately, that is not the reality. On average, only 50% of the people can benefit from drug treatment. If you are unlucky to be a cancer patient, the chance of you getting benefit from the treatment is only 25%. That means 75% of the patients, they don't get any benefit by taking a drug. The reason is we don't know how to differentiate a good patient from a bad patient. A good patient are the patient who benefit from treatment. They get good benefit efficacy. They don't get side effect. But unfortunately, in the current situation, a lot of people, not only they don't get any treatment benefit, they also get serious adverse drug reactions. I don't think anyone wants to be one of these patients. They don't get the benefit, instead get a side effect. Because of this, that's really because we don't know how to tell one patient from another patient in the entire current procedure of drug development, from a discovery to preclinical based animal testing, then through phase 1, phase 2, phase 3 clinical trials. This current procedure, we develop drugs. It's long, costs a lot of money, takes over 10 years. And as chemists like me, we need to synthesize about 10,000 compounds before a lucky one can get through all the process, then get regulatory approval by the US Food and Drug Administration or China FDA. That means the success to drug discovery is rare. That's why the drugs are so expensive, especially those innovative drugs. And in addition to the difficulty of making a drug to the market, from discovery to market, even the drug is approved, there is still a chance where side effect will happen. Why that case? Because in the clinical trials, only dozens, hundreds of patients are included in the clinical trial, but after the drug is approved, hundreds of thousands of patients, even millions of patients will take the drug. There is a problem. Whether the hundreds of patients in the clinical trial can represent the millions of patients in the future to take the drug, that is an unknown problem. That's why, even though the regulatory agents like the US FDA, China FDA did whatever they can, but serious adverse drug reactions still take place after the drug is approved. It's not because the regulatory agents did not do a good job. It's just because humans like us, we are quite different from one to another. This slide shows the failure happened mostly in the clinical trials. Putting a drug, after all the animal testing, we demonstrate the drug is efficacious in animal models and safe for animals. Then you give a big package of paperwork to the agency, and you say, my drug has been tested safely and effective on animal models, and you may be able to lucky enough to convince the agency to initiate clinical trials. That's what we call phase 1. First in man test of your drug candidates in human beings. However, the successful rate is extremely low. You only get if you are working in the cancer area, you only get 5% of the clinical candidates to be approved. That means the other 95% of the candidates you put on human clinical trials will fail before you find that lucky one. That's why the drug discovery process is so expensive, especially during clinical trials. The reason is we don't know how to differentiate one compound from another compound. If we have better ways to find that compound, which will eventually be able to go through all the screening process to demonstrate its efficacy and safety, then we will be lucky because the development process will be much shorter and the expenses to develop the drug will be much lower. That's why 16 years ago, I went back from the US to China, tried to set up a drug discovery company with a few friends, including this gentleman, Dr. Lu. He's still in that company. But after several years, I left the company because my wife asked me to go back to the United States. But we are lucky enough because after 12 years, we were able to put this drug, a anti-cancer drug, to be approved by China FDA, and clinical trials are still ongoing in the US, Japan, and other countries. And currently, this drug has been saving thousands of patients' lives each year. It's quite gratifying for myself. My work 16 years ago, eventually, were able to benefit people. The approach we use differently with other companies is called chemical genomics. That is to look at the different interaction between chemical candidates with our individual genomic information and see how this chemical and genomic interaction really differs from one patient to another. That's why we were able to do this. And if you are lucky, you have your compound or selected your candidate for clinical trials, there is two possibilities. One, some patients benefit from your treatment. Some other patients, they don't. They even develop side effects. So the question is, can we do something to tell the difference between the two groups of patients? What makes them respond to the same drug differently, although they have the same disease or symptom? This is what my field we call precision medicine nowadays, and previously we called pharmacogenomics or tailor-made medicine. We are trying to collect a lot of omics information about each individual patient, and we use a lot of statistical method, machine learning method, artificial intelligence method, trying to correlate the relationship between our omic information and the treatment outcome to build a model. This model, if it works, we can use the model to predict a future patient whether he or she will respond to the treatment favorably or not. If the patient is predicted to belong to the group not responding well or even develop side effect, then we should not give the drug to that patient. Or the patient who gets the drug will have good response and will not have side effect. That's the general concept of precision medicine or tailor-made medicine. But the idea is, the challenge is, this model, or what we call a biomarker, needs to be predictive. That means you can make very accurate prediction. We can do this not only for pharmaceuticals, but also radiotherapy, even for traditional Chinese or traditional Chile medicine. We have a lot of different ways to describe ourselves in terms of our genome, DNA, and our RNA transcriptome, and how the proteins work and express in different quantities. And even we can look at their metabolome. So we can easily generate billions of data points to describe each and every patient in a clinical trial or in the clinic. That creates a lot of opportunities and challenges. And the opportunity will allow us to investigate how individual chemical structure interact with the human being. The human being is being described by billions of data points so that we can find out their relationship, the match between the chemical structure and the personal genomic information. In our field, we made a lot of progress based on individual cancer patient genetic profile, a specific gene mutation, or our expression of particular gene. Then drugs have been made against those particular patients. You can see that list going longer and longer each year. That's great. And we can also use this information to eliminate the occurrence of side effects. That's some of the examples the US FDA have approved. However, our field have not been very productive. We really need more biomarkers, but we spend a lot of money, but we have not been able to generate enough biomarkers for all the drugs, thousands of drugs approved by the US FDA. This is the question, why our productivity in terms of the number of new drugs and the number of new biomarkers have not been as many as we really like to have. The reason, one of the reasons is we lack reproducibility in our field, especially to quantify and characterize genomic information. That if we don't have good omic data, as the X shows, even though we have good phenotypic information, there is no hope we can find good biomarkers. We only need to make one mistake in the entire process. I just want to show you some of the ugly examples, why and how irreproducible our field is. The sentence should be 100%, but it's only 4. And here it should be 100% in the middle. The same data set analyzed by 2 groups should be almost the same, but in reality, they're only 20% the same. The other 80% of the data, the result we get, are completely depend on who analyzes the data. That's a big problem. And now we use patient's DNA to sequence them, to find the mutation, and prescribe a targeted therapy. But our concordance using 2 different approaches is only 25%. That means whoever took the drug is largely dependent on where we send our samples to be analyzed, generate the data, and interpret in a clinical way. So there are too many ways to analyze the data, but we really don't know which way is better. So over the past 15 years, we spent a lot of time engaging our 100 organizations, 300 scientists to look into this issue. I don't have time to go into detail, but you have to believe with me. We worked very hard, found out the underlying problem in the field. What we found out is, with the same set of samples, who generates the data make a difference, who analyzes the data make a difference. We really need to have good standard operating procedure and quality controls. So that's why we are generating a lot of reference materials, reference data sets, so that we can evaluate the data quality and evaluate the performance of different data analysis approach so that our result can be reproduced. So that's the overall structure, how to make precision medicine a reality. We need to have good biospecimen. We need to collect good phenotypic information for those patients and different types of omic data from genomic, transcriptome, proteomics, metabolomics, and the metagenome data, and so on. Genetic data in high quality. Then all this big data analyzed in relationship to the phenotypic data like treatment response, then this kind of big data mining will be very useful for us to find new biomarkers for diagnosis, prognosis, treatment selection, and to predict and whether resistance will happen, and even better, to prevent diseases from happening by using preventive measures. So each and every step needs to be quality controlled. And China has been working towards this direction very hard, and the new results will be coming in the next few years, so that when that day comes, I think we can do much better job, have a precise, tailor-made medicine for each and every individual. And we are not working alone. We are working internationally with a consortium, now an international society, trying to, with the goal of enhancing the reproducibility of high-throughput technology, so that we can better and more accurately describe our individuals, so that we can prescribe new drug for each and every individual patient. That's all I want to share with you, and thank you very much. Gracias.